minds.
All right, I'm going to turn on screen share while we do this.
All right.
Then I'm ready to
Welcome,
move.
everyone, to the EVOS seminar series. Our guest today is Dr. Terrence Deacon, who's going to be giving a really interesting presentation on the hierarchic transitions in evolution. So I will leave it to him.
All right. Hello, all. I'm going to start right in with just a very brief statement, and then start in. And while I'm doing it, I'm going to bring up the slides I want to talk about. What I'm going to say might initially sound like it's sort of anti-Darwinian in some sense. It is not. I am fully within the fold, so to speak, of Darwinian evolutionary theory. This is basically an attempt to expand it without the sort of polemic that's been happening over the last decade or so, where people are saying something is wrong with the theory. Basically, I think nothing is fundamentally wrong with it. It just doesn't deal with all that's relevant, including, for example, evolutionary theory is completely agnostic about mechanism, and that's basically what we've been doing over the last 150 years, is figuring out sort of the mechanistic nuts and bolts of evolutionary theory. And so this could be considered as sort of a nuts and bolts edition, which says that there's one aspect of the evolutionary process, difficult to explain in simple Darwinian terms, that I think actually requires including what would be almost the opposite of a natural selection effect. And I think that that's responsible for what I call hierarchic transitions. So that's what I want to talk about in a nutshell. But it's the nuts and bolts I want to get into. So let me just move right ahead. One of the things that has contributed to my thinking about this has to do with an argument that was finally put into words by Urs Sathmary and John Maynard Smith. John Maynard Smith being one of the sort of key names in late 20th century evolutionary theory, basically trying to follow the standard model. They both began to realize this difficulty with what they called major transitions. And I'm going to ask the question here, what makes a transition major? I actually have known Urs Sathmary for decades now, and he and I argue about this on and off. But I don't see all of these transitions as major, and in fact, I disagree with their approach to this, which is ultimately an approach that could be characterized by Richard Dawkins' notion of replicator selection. Urs, like John Maynard Smith, actually had that as his driving logic for understanding how evolution works. I think that it's a small part of the story, and that the replication model actually hides a lot of what I would call the thermodynamics of evolutionary processes. That is, how things get generated, not just how things that are generated can vary and then be selected, but actually the very process of generating structure and function, which is basically ignored. As I said, it's in effect the part of evolutionary theory that's just sort of taken for granted and not analyzed. In any case, I've underlined on this list of their 8 major transitions, I've underlined just a few that I want to talk about today, because they're the clearly hierarchic ones. Prokaryotic to eukaryotic cell transition, moving from single-celled protists to animals, plants, and fungi, the movement from individual organisms that, in a sense, are not social to not only social animals, but eusocial animals, or like us. I think that we human beings are what someone has dubbed ultrasocial, as we can be socially interdependent in the millions. And then finally, an issue that was driving my research since the 1980s, the problem of the origins of language, and how that, in effect, is also a kind of hierarchic transition. I'll come to that at the end. A book that really caught me to focus on this was written back in the early 80s by a man named Leo Buss, and I recommend it to all of you. It's called The Evolution of Individuality. You might think this is just the opposite of the sociality problem I want to talk about, but he was basically talking about how it is that a multicellularity evolved. We've learned a lot more since then. But Leo, coming from a sort of standard evolutionary perspective, recognized that in the three major lineages of multicelled organisms, fungi, plants, and animals, they all had one of the same problems that they had to solve. And they solved it in different ways, which gives rise to the diversity of each of these groups. And what I want to point out to, for those of you that know something about the history of social theory, and particularly social theory of justice, the philosophy of justice, John Rawls' theory about how a just society is produced involves what he calls a veil of ignorance, in which individuals don't have foreknowledge of where in society they will show up, at what level of society they'll show up, and so on. This veil of ignorance is how he describes it. It's a sort of a thought experiment. And the idea is, if you don't know where you're going to show up in society as you pop into the world, but you have the ability to decide on what kind of society to set up, not knowing where you will be, you can't, in a sense, put yourself in the best spot. So one of the things you want to do is to create the most just society possible. So that when you are randomly assigned some role in the hierarchy of society, you have the best possible chances. Well, to some extent, Leo Bus says that's what causes these three different groups. And I want to just sort of summarize very quickly what he says. And he says that what has to happen is that individual cells cannot have, at the beginning of an organism's life cycle, individual cells cannot have the ability to determine where they will be. That is, they cannot, in particular, determine in a way that makes it possible for them to become the reproductive cells. That is, the ones that will persist forever. That is, will replicate, and they'll have offspring and be around forever. If that was true, he says, then in effect, multicellularity would fall apart. And cells in organisms that do this become, in effect, cancer cells. That is, they take back autonomy and they begin to reproduce irrespective of the whole, and typically that destroys the whole. So he said the way that this happens is that each of these lineages has to create, in effect, a veil of ignorance for the cells. And the three groups I show here that he describes, fungi, plants, and animals, do this differently, and this accounts for their different kinds of organization and what they can and can't do. So in fungi, each nucleus of the cell produces proteins and RNA and so on, but the membranes in fungi are somewhat porous, much more so than in these other two groups. And as a result, you can't be sure, that is, a nucleus, a set of genes, a genome, can't be sure that you're controlling only the surround of your nucleus rather than neighboring cells. And that means that, in a sense, neighboring cells are always in each other's business. That makes it impossible to sort of guarantee that you, local nucleus, can be the one that reproduces. It can determine the next stages so that you're always in the fruiting body. And as a result, fungi have this very sort of ubiquitously same cell type throughout, so that the rhizoids beneath the ground or the fruiting bodies above the ground all effectively are the same cell type. There's not a lot of what we call tissue differentiation in fungi. It's why all parts of the mushroom taste the same, so to speak. So this is one of the ways to create the veil of ignorance, so to speak, at the level of cells in a multicelled organism. Plants do it differently. Plants do it by creating cell walls that make it impossible for cells to determine where they are, to move around into the ideal places. So, for example, a cell can't determine that it's going to be an apical meristem that will eventually produce seeds. It's impossible because you're stuck in place. And you're born, you're stuck in place, you can't move around. You have to wait until someone else down the line becomes the reproducer. So in effect, that's a different kind of veil of ignorance. You can't move under those circumstances. And finally, with animals, interestingly enough, what happens is that the ovum, in a sense, mother's DNA in the ovum generates a whole lot of protein and RNA. In the unfertilized cell, the sperm fertilizes it. Now the genome of the newly fertilized egg, the zygote, is in effect going to go through the first large number of its cell divisions, totally under control of mother's genes. That is before the fertilization took place. And what that does is it locks in cell lineages into what's going to be their future fate, so that in effect, no cell has the ability after fertilization to determine that it's going to be a reproductive cell, a sex cell, a gene cell, gene-carrying cell in the ovary or in the testes or whatever in the organism. So these are just three different strategies, all producing a different sort of logic of veil of ignorance. This caught my attention. These are very clearly Darwinian arguments, but it's about how do you create a just society, a just social group in which everybody will tend to cooperate, so to speak. And this kind of logic, of course, extends all the way to talking about social processes, even in all these theories about social altruism. In fact, it's the classic altruistic story. One of the things that's important in this is that this, as a result, it limits the autonomy of the units that are involved. And of course, there's other ways to do this and use sociality in ants and bees. One of the things you do is, in effect, this is limited by genetic similarity, and reproduction is controlled by a queen, by a single individual in this group. And as a result, what we see here on the top left is ants, worker ants, who actually extrude eggs to feed the larva of the queen. In other words, their reproduction is literally a reproduction in order to help somebody else's reproduction. Down here in the bottom left, honeypot ants, a subset of the workers basically attach themselves to the roof of their colony and allow themselves to get fed and become basically containers for holding on to honey that will keep the colony alive during difficult times. These individuals will never reproduce, will never forage, and effectively will eventually die as honeypot containers. And of course, in termites, we get this incredible diversity of body types, including this queen here with this massive abdomen filled with eggs. She can't move herself. She has lots of helpers that help her move around, feed her. She have to keep moving her so that her tissues aren't all sort of weighed down by this process. She can't give birth on her own without help. She can't feed without help, and so on. Termites are not haplodiploid like ants and bees, but they're highly inbred. And so all of this has to do with, in effect, using, in this case, genetics, which plays a role in aiding, or you might say biasing, this group towards a loss of lower order autonomy. Individuals can't pass on their genes by themselves any longer. So it's a different variant of the same problem, but at a higher level. So this leads me to what I call the paradox of higher order control, which is what's going on here. There's a higher order control in which sort of like you might think of it as top-down control of the group, in which lower order elements, whether cells or individual organisms, individual ants, are sort of dependent upon the higher order control. So the evolution of hierarchic complexity requires that lower level mechanisms lose autonomy of function and instead contribute to the higher level functions. Loss of specificity and degradation of the function of lower level units can aid the emergence of synergistic complexity by enabling the interactions of their incidental features to be expressed. And I'll come back to the expression of what I call incidental features in a few minutes. But sacrificing function is dangerous without the ability to anticipate and assess the probability that higher level synergies will develop and be effective. In other words, we human beings can do this because we can talk about it and sort of plan for the higher order processes to do what we want. But of course, in evolution, there is no forethought, no planning. In effect, how do you lose autonomy in a safe way? In effect, is the question we're asking here. Anticipation, prediction, and social agreement are unavailable to biological evolution. And they're difficult to achieve even in human society, as we know now in the United States. So what I want to do is to talk about this process from the bottom to the top. I want to start at the lowest, not maybe the lowest level, but a very low level, at the level of genes themselves. How do you get synergy evolving among genes? Now, obviously, in the replicator story, what you want are genes that replicate themselves. But in effect, if you've got a lot of synergistic relations, then you can't replicate yourself independent of anybody else. In a sense, you need everybody's help. But what this means is that local replication, that is individual replication, the selfishness of the genes, if you will, has to be to some extent lost or sacrificed. There has to be some loss of autonomy. And so in this first model, which is just based upon the notion of gene duplication, I want to identify a general logic that I'll follow all the way up. And I list this at the bottom. So gene duplication is probably the most common form of genetic mutation, in which simply during crossover or by virtue of viral infections, or simply mobile elements in the genome that in effect move along with themselves large chunks of the chromosome. So chromosomes can have sections deleted, or they can have sections duplicated, and this is a duplication. Typically, this kind of duplication might be the result of an uneven crossover effect during meiosis. But the general logic I want to get to is that if you start out with duplication of a functional unit, something that has some autonomous functional output, it produces a redundancy of that function. And having redundant function relaxes selection on each of the redundant elements. That is, natural selection becomes less effective. And this will lead to effective degradation. That is, when natural selection is relaxed, what you're getting is a loss of purification effects, so that you're not eliminating errors at quite the same rate. Now, in sexually reproducing organisms, this has an interesting consequence. That is, if you get independent degradation in different duplicates, you'll also get random sampling of synergistic combinations, or rather random sampling of combinations, some of which might have synergistic effects, some of which won't. The aid here is that because of the redundancy of function, synergy becomes more likely. And I'll come to that in a few minutes. Consequently, however, if by recombination a synergistic outcome actually is an aid to reproduction, then in effect there's a new stabilizing effect that will show up, again, fairly standard Darwinian selection story here, at the end, that will stabilize this new combination, but stabilize it now into a more synergistic effect. So let me give a sort of really simplistic sort of way to look at this. So in this model here, this is supposedly the chromosome. Here's the coding segment of gene. It produces a protein. I will call that phenotype sub 1. And then by interaction with lots of other aspects of the cell, of the organism, and with other proteins and so on, and other molecules, eventually it produces a phenotype N, which is some function. And so I diagram it in a simple sense here. A function that might be a chemical reaction that has a certain affinity and specificity for interacting with other molecules. Under gene duplication, however, so here's a duplication event. One of the things that happens is that now you have the same gene producing the function twice. If the function of having more of this stuff produced is troublesome, deleterious, then one of the things that will happen is there'll be selection favoring damage to this gene so that it no longer produces a product, but oftentimes slight damage will produce variation, just slight variation in its functional consequence. And here I show it as sort of 3 different curves, each of these being in some individual in the population, one of these variants. So what's happening is the original function is preserved because of the redundancy, and now you get sort of a random walk of alternatives that are just slightly off or variant from the original function. But if this slightly variant function actually produces something useful, that is by having 2 slightly variant versions of the same thing, you can do sort of more than one thing. You can have your cake and eat it too, so to speak. Then a new synergy can arise. And what happens when the synergy arises is that it also changes selection on the original. And under these circumstances, the random recombination that in effect just hits upon this, because now you have a bunch of individuals with slight variant versions of this gene in the population, the 2 can begin to complement one another. And if they complement one another, this will re-expose the copied gene to selection that also modifies selection on the paralog, that is the original gene here. Both of them will now be selected not just for the function, but for how they complement each other. That is, there will be a level of selection that's now not just on the individual, but it's distributed across 2 or more variants. Probably the simplest example of this has to do with the gene for producing hemoglobin. We know, for example, that the ancestral hemoglobin gene was also a duplicate with a common ancestor with the ancestral myoglobin gene, the gene that's within, for example, red muscle cells that binds oxygen, red for the same reason the blood is red, because of the iron molecule that's bound. But the hemoglobin molecule itself, now floating in blood, not within fixed cells in the body, also has gone through a duplication process in the evolution of animals. And that duplication has produced initially an alpha and beta variant that's in all mammals, for example. And the 2 variants of the gene each produce a slightly different hemoglobin molecule. This lozenge-like structure here is what we call a porphyrin ring. It allows this protein molecule to suspend an iron atom right in the middle. Iron is very reactive, and so keeping it away from the rest of the protein is important because it would deform the protein as it becomes oxygenated, that is, as it effectively rusts. What's happened in the evolution of hemoglobin is that the hemoglobin in your blood and my blood is actually made up of each molecule is now paired with another alpha and also paired with 2 betas. What's happened is in their independent evolution, at some stages, the back sides of this molecule, that is the parts of the coding gene that describe the sequence of amino acids, did not change much around the oxygen binding element because it has to be very conserved here in order to maintain that iron atom. But in effect, it was less conserved on the backside, so to speak. And as a result, this allowed complementation of structure on the back sides of this, so that ultimately in your blood and my blood, alpha and beta became a synergistic in terms of their physical structure and their tendency to bind together. The result is that they tend to stick to each other because of sort of hand-in-glove kind of features of their, you might say, the backside away from the oxygen element. And that produces a kind of tetrahedral molecule in which there is now not just 1 iron atom, but 4 at opposite ends of the tetrahedron, maximally apart from each other, can carry much more oxygen. So in effect, a new synergy evolved with the duplication and variation of alpha and beta with respect to each other. But the hemoglobin story is even more interesting in the sense that, in fact, the beta itself also has duplicated into a series. And here we see that series all along chromosome 11 in humans. each one of the blue ones actually produces an actual hemoglobin molecule. Whereas these two grays are what we call pseudogenes. That is, they've accumulated errors that cause them not actually to be coding. They no longer produce a series, no longer in fact produce even RNA. So in effect, you only get the production of these. So duplication doesn't always produce something functional. It oftentimes produces something that just simply has enough of an error in it that it no longer does anything useful. But what's interesting about the duplication of the betas is that, in effect, they're read during development in this direction, from epsilon to gamma to delta to beta. And this has happened in placental mammals. And the story is that the fetus in the embryo inside a developing mother has to get its oxygen from mother's hemoglobin. The result is that in the early stages, when the placenta is poorly developed, and in fact the embryo is not producing a lot of hemoglobin yet, you need to have a fairly strong binding capacity to get enough oxygen from the hemoglobin in mother's blood. As a result, in early stages, epsilon is produced. And epsilon has a much higher oxygen binding capacity than standard beta. Later on, during most of gestation, it's one or the other of these gammas produced in sequence that also then replace epsilon as the fetus gets better at doing what it does. But of course, after birth, one of the things that has to happen is you have to change your strategy altogether. To have a really high binding capacity of the hemoglobin in your blood is great if you have to steal oxygen from mother's hemoglobin, but it's bad news in handing off oxygen to your own myoglobin in your tissues. And so once you leave the womb and you don't have to steal oxygen from mom, so to speak, it now becomes useful to shift that oxygen balance so that you're not so highly bound to it, so it's easier to give off to your own tissue. And so as a result, what we see is a synergy across time in which the duplications have produced a sort of interaction relationship that produces this higher order synergy across time. When each one of these is not sort of doing the whole story, each one of them hands it off to the other at a particular time. So it's a higher order synergy, a relationship between these. You might say it's a cooperative relationship in some abstract sense. There are lots of ways that this can happen. I won't go through this, but in a sense, it's one of the most common features for building complexity of function in the body. So for example, the acetylcholine receptor, a transmembrane complex made up of 5 different proteins, variants of the same acetylcholine molecule, acetylcholine binding molecule, a complicated protein with a transmembrane section here that allows it to fit and cross through the membrane. And the 5 of them together create this pore, this tube that has in the middle, although it doesn't show it here, a kind of gateway that opens and closes when acetylcholine binds to it and allowing ions to flow through. But this is made by having slight variants of the binding molecule produced here. And these variants, there's 3 different variants here, all linked together in such a way that it makes it possible for this to form, you might say, a cooperative complex. Couldn't have been done with just one of them, but it also couldn't have been done with just one of them that is identical. A similar example for steroid hormones like testosterone, estrogen, cortisol, and so on, bound by a receptor molecule that carries the now complex of steroid and protein into the genome and activates genes. There's a conserved segment in each of these binding proteins. So here we see growth hormone, we see androgen hormones, a variety of them. What we see is that there's a conserved element, and these are the parts that have to do with the DNA binding. There's slight difference in terms of these sort of tails here. But this is the result of duplicating a receptor molecule many, many times. So it's now possible not to just bind to one kind of steroid hormone, but now multiple steroid hormones. The other thing I want to focus on, which is going to come up in just a second, is that you're not only able to duplicate coding elements. Here's the coding element of this gene in this sort of purple part here, but you also have leading elements, which in effect are promoters or inhibitors that are very distinctive structures, the result of the sequence of base pairs here that's very specific and allows certain molecules to bind to this and to control the release and the coding or stop coding or the rate of coding of this coding segment. And it's those sections that can also be modified. So we see here modification, elimination of some coding or inhibiting site, as well as changes in the coding segment. So you can get regulation of when a gene is produced, regulation of when a particular variant is produced. And this is, of course, one of the things that's relevant to what I was just describing in terms of the timing of beta hemoglobin production and how it changes over time. You have different coding segments that then sort of activate or inactivate the gene. So I want to point out that this feature actually is relevant to another question, and a question that one of my mentors from way back, Gregory Bateson, suggested in a book of his called Mind in Nature, in which he asks the question, what pattern connects the crab to the lobster and the orchid to the primrose and all four of them to me and me to you? And what he's getting at is the theme and variation logic that makes up multi-celled organisms like plants and animals, and as I said, doesn't do a very good job of making up this sort of differentiation of cell types when we look at fungi. But in animals, for example, the lobster and the crab, have similar sort of appendages, but laid out quite differently. There's a lot of commonality when we compare the two, but there's also commonality when we compare within the organism. So all of these legs of the crab look similar. These legs in the lobster look similar. They actually have a correspondence at a higher level to the crab. And of course, each of them has a pincer towards the front, which is also a modification. You'll notice there's little pincers on the end of the lobster secondary claws here that becomes heavily enlarged in both the crab and the lobster, so that there's a similarity within the organism of body parts, duplication and synergy between these, and then a relationship of homology between different groups here. We see the same thing in plants. In fact, this was one of the first things identified at the beginning of the 19th century by Wolfgang von Goethe, who looked at plants and said, hey, isn't it interesting that the distribution of leaves on a flowering plant and the distribution of petals follows a similar logic. And the structure is a kind of theme and variation. He basically made the argument that maybe the secret to life is understanding this sort of theme and variation structure throughout the body. But of course, we see the same kind of thing when we compare different plants like the orchid and the primrose. So there's a similarity between the petals, but there's also a similarity between petals and leaves. There's also a similarity between stamen and stems. These are driven by different kinds of genetic effects, but in effect, they're the same kind of effect in the sense that there's a gene duplication effect, there's a regulatory effect on the expression of genes that makes this possible in multicelled organisms, and that's the result of duplication of these regulatory elements. So this theme and variation is really critical to building complicated multicelled bodies in which you've got synergies within the body at the level of whole organs. And we learned in the 1980s that this is in fact the result of another kind of gene duplication. Duplication of a gene that produces proteins that don't go and produce other proteins or don't interact with other proteins in the body, but instead bind to these promoter or inhibitory elements that drive other gene expression. Now, if these have similar binding characteristics, a single gene that produces a single protein can now affect, maybe slightly differently, the expression of multiple other genes. We typically call this a regulatory gene, although there's lots of other ways to talk about it, but basically a gene that produces a protein that controls the expression of other genes. And of course, one of these could also be a regulatory gene. So you can get a hierarchy, a sort of cascade of these effects. Well, in the 1980s, one of the first things that was discovered looking in fruit flies is that, in effect, one type of these regulatory genes, called a homeotic gene, or a homeobox gene, or now called Hox genes in other animals, itself was duplicated. So if the regulatory gene gets duplicated, now one of the interesting things is you'll get theme and variation effects in terms of what downstream genes they turn on and off, and how they do so. I like to think about this as sort of like a conductor who gets, put in sort of, think of a Star Trek example. The conductor is put into a transporter or sent off somewhere, but the original conductor is not eliminated. So now you get a sort of a duplication of the conductor. But they have different sort of life history experiences over time. And the way they conduct the symphony of other genes, in this case, will now vary slightly. And you'll get theme and variation. The way they conduct the same effects will now have a theme and variation logic. But it turns out that in these Hox genes, these homeotic genes, genes that bind to other genes, genes that produce proteins that bind to other genes, have in fruit flies and in most animals, have duplicated themselves many, many times. So now you don't just get one duplication, you get a large number of duplications. And it turns out, so here you see the color coding here on the fruit fly. It turns out that they're expressed not over time alone, but also in different parts of the body. So here you see the embryo and the expression when these genes get turned on in which parts of the body. And as a result, it produces segmentation of the body. And one of the things about many, many animal species, the bilaterality in general, is that they're segmented in a sort of longitudinal way. And you can see in fish, in mammals, the same sort of logic is played out. But one of the things that's happened in most vertebrate animals, surprisingly, is it's not just that the sequence of duplicated genes is a sequence that's duplicated, but actually the whole sequence is duplicated. We think one of the things that's happened is that there's been actually whole genome duplication. So you can see one duplication, and then the duplication is duplicated. So this is this incredible complex that we can see. And then you can see some of them are turned off, some of them are turned on, some of them are turned into pseudogenes, some not. But this allows you to produce this complexity of body form. So what I want to point out is that this is similar in many respects to the kind of thing that goes on even with a simple duplication, like I showed you before. And it's responsible for many of the diverse forms that we see in lots of different animal groups. And of course, to produce some of these really complicated organisms like vertebrates, but also insects and so on. You can get really complex results as a result of turning things on and off, duplicating more, knocking out some, and so forth. I won't go into all of that except just to point out that this helps explain this theme and variation logic of the body. Because if they're all producing similar segments, like in this millipede and centipede here, it's a sort of theme and variation. They're all slightly variant, but they're very, very similar. But in fact, what we have the possibility of is that now gene expression differences can take on a theme and variation effect at the level of whole phenotypes, whole organism body parts. So that what we see is that the different body parts, these different segments, now can evolve with respect to each other. That is, by virtue, again, of sexual recombination and looking at combinatorial effects, you now have the ability to enlarge some, shrink others, so that the antenna parts, the mouth parts, the spinnerets that make the web and the spider, and the parts that give, allow birth of the egg, production of the egg, and so on, all are using the same theme and variation logic, but now it's a logic that can be expressed at the level of whole bodies. So that this theme and variation logic that is the result of duplication and dedifferentiation, that is loss of some function, now allows these higher order synergies to evolve. And you can see that it particularly easily seen in these segmental animals, again, like lobsters or insects or spiders. And you can see these incredible variations, so that, for example, in the tail of the lobster, you've got these little swimmerets that are fanning water over their oxygen sort of gill-like structures that play a very significant role in oxygenating this animal. But at the same time, you have reproductive elements that are made up of the themen variation. You have the claws that are made up of it. The antennae are using the same feature as themen variation. So what happens is that by varying now this sort of secondary hierarchic effect, so you vary the controllers of duplicate genes, you now have a sort of higher order duplication that can produce this sort of synergy at the level of whole body parts. So what you can see from this is that I'm beginning to build up a story about how you get higher order synergies using the same kind of logic again and again. Let me now shift a little bit to show how it can be even more complex in its hierarchy. What happens if the duplication effect, that is the functional consequence, is the result of something in the environment? Something in the environment does what a gene might do, or does what the function replaces the function. Under these circumstances, the reduction of selection that results is not because there's a duplicate gene that's doing it, or not because something else within the body is doing it, but now something outside the body is doing it. And that will, in a sense, reduce selection and allow dedifferentiation of the gene that's maybe producing this protein. So extrinsic duplication also produces this same effect. I oftentimes describe this as masking. It masks selection so that it allows variation to accumulate. So long as this external force factor is out there, it will allow a whole lineage potentially to have a degenerate genetic effect. Interestingly enough, because it's extrinsic, because it's not always available in the same amount exactly in each individual at each generation, there is now some increased selection. Once this function has degraded, there's now selection to maintain this extrinsic source. If it allows degradation sufficiently so that this, in effect, has no consequence, a pseudogene, like I showed you with the hemoglobin beta example, now it's the external source that's providing the function. But this means that any other locus within the genome that maintains or guarantees or makes it much more likely that this external factor will be available are now going to come under selection. This is now under selection not for producing its original function, but a function that may play some indirect, not very precise role in helping maintain increasing the probability that this extrinsic source will stay constantly available. So now other loci in the genome become under selection for a function that was not what they originally evolved for. And because this is a distribution effect, it redistributes selection that was simply located on maybe one gene, but now you get redistribution of selection across the genome on any loci that have any role to play in increasing the probability of maintaining this extrinsic function. So what's happening now is extrinsic redundancy forces a kind of redistribution of selection across the entire genome, and with respect to a single extrinsic effect. In other words, now a new kind of synergy is developing. It's a very partial synergy, so that each of these other genes and their consequences, their functional output, is partially now being used for something else, but collectively. So their collective effects, that is, their synergistic effects, become more appropriate. What this says is, in effect, what was, in a sense, determined by a single gene, by a single locus, is now distributed in such a way that if perhaps out of the dozens of loci that might be affecting this, if one of them degrades a little bit or varies a little bit, it's multi-stable, or what some people call metastability. That is, you have multiple loci that maintain it, multiple ways that it can be varied, but it's also more stable in this respect. But it produces relationships between genes that may be quite separate with quite different functions that didn't exist before. Again, higher order ones. So a simple example of an external functional duplication occurred with respect to the synthesis of ascorbic acid, vitamin C. What we know from really beginning in the 1950s is that, for the most part, most other mammals and most other animals in general, including fish even, produce their own ascorbic acid. It's produced endogenously, genetically, except that Old World monkeys, New World monkeys, chimpanzees, apes, and humans don't. We need to eat something that produces vitamin C in order to have it. Now, I should say that there are other species that in fact need to eat vitamin C. Many birds need to do so. Some guinea pigs need to do so. Fruit bats need to do so. These are all hints as to why we might need to also eat foods with vitamin C. But in effect, all monkeys and apes have to regularly get ascorbic acid in their diet. In the early 1990s, 1994 to be explicit, a group in Japan looking in the rat genome identified and cloned the gene that produces the final step in synthesizing ascorbic acid endogenously in the cells of rats. Now, what's interesting about this is that once you've cloned this gene, you can use that clone, tag it with a radioactive or a fluorescent tracer, and look for its presence or its similarity to genes in other organisms. And they did so initially, in fact, with humans. And what they found is that the rat gene, sometimes called GULO or LGO, as short for L-gamma-lactone oxidase, this final enzyme in the step of producing ascorbic acid. Actually, it's produced ultimately from glucose. Here are the exons, that is the coding segments, separated by introns in the rat genome. And it turns out that in humans, we have corresponding exons for a number of these. Not all of them, interestingly enough. Some are deleted. Each of these points are, in effect, places that we see mutations, SNPs, single nucleotide variations. Those that are marked in red here, this is supposed to be stop signs. These are stop codons that would cause the RNA molecule to sort of deconnect from this gene if it was connected. But in fact, none of this stuff works because the whole thing has received a frame shift, because upstream from this, a whole unit was knocked off, a whole cross-link of the DNA has been knocked off. So a frame shift is like if you shifted your movie projector out by just a fraction of a frame, so the frame is always broken up. It turns out that this would produce, if it produced anything, would produce nonsense. And it turns out, now that we've looked at other primates, all other primates have some variant of a knocked out gene for producing ascorbic acid, which says that at one time it did exist, but stopped existing, so to speak. It became masked and degraded. The logic of this, I think, is pretty straightforward. We begin with normally endogenously producing gene that's in the genome, but somewhere around 35 million years ago, the ancestors of monkeys and apes began to eat fruit. They were, as we can tell, for the most part, nocturnal insectivores before this, and had to produce their own vitamin C. But fruit maintains its edibility for birds initially, and now for primates and other species, by having lots of antioxidant in it. And the antioxidant is vitamin C, ascorbic acid. But as we begin to regularly eat these sources of food, they're also found in the trees where insects were, we begin to have external duplication of this function. The gene can now acquire certain mutations. At some point, if enough mutations are acquired, as I mentioned before, selection is now distributed to other ports in the genome that make it more likely that we'll constantly have availability of fruit. And what are some of those examples? Well, one of those has to do with color vision. It's only primates, anthropoid primates in specific, that have developed three-color vision. Our ancestors, to all land mammals, in fact, all aquatic mammals as well, the ancestors were all nocturnal and had a nocturnal vision, and therefore not three-color vision, but elaborated sensitivity. Here, this gray curve here represents the sort of light sensitivity of the rods, which was roughly the light sensitivity of what subsequently became cones that have lost their sensitivity for certain frequencies. Here's the blue, the green, yellow, and the red. And this is because of another duplication effect. As on the X chromosome, it turns out that the colored pigments, the rhodopsins, have itself, that molecule has been duplicated, and a random walk, so to speak, has caused them to lose some of their sensitivity, but their variation. And it's done so slightly different in Old and New World monkeys, which is shown here by this diagram. I won't go into the details. But being able to distinguish color change, specifically change from green, like in leaves, where the fruit is easily, you might say, camouflaged in the tree, to a color change of a bright contrasting color like blue or yellow or red, suddenly becomes relevant because it tells you that now the fruit is edible. So having, again, a synergy of color vision developed in order to pick out ripe fruit becomes necessary when you need to regularly get fruit. There are many other aspects of the primate physiology that were relevant to this issue. For example, we have a particular modified glucose transporter in our blood that does a good job of also transporting vitamin C in the blood from one place to another in cells. Not necessary if you produce your vitamin C endogenous to cells. So there are many things happening, but my claim here is that, in effect, this duplication effect produced now this change of the body, in which there are lots of other aspects of the body that after degeneration took it up in a more complicated way. This kind of logic is also relevant to the evolution of eukaryotic cells from prokaryotic cells, that is, bacterial-like cells. And one of the things that we've learned over the last half century is that the mitochondria and the chloroplasts that are in plants, and the mitochondria in plants and animals, is the result of, in a sense, invagination, in which a bacterial ancestor was taken up inside of another cell type. And each of these, because they had a bacterial-like ancestor, a plastid and a regular prokaryotic bacterium, each have their own genomes. So what happens is that there is chloroplast genes, there are mitochondrial genes, and there are nuclear genes in sort of plant cells, and mitochondrial genes and nuclear genes in animal cells. But what's happened in this process is that these are, in effect, external. Like the vitamin C case, these are extrinsic sources of information, extrinsic genes, compared to this. So it's like relationships between organisms, but now very different organisms. What we've learned from this is that, in fact, the ancestors of mitochondria had roughly 5,000 distinct genes, which is typical for most other bacteria from this lineage. Chloroplasts, which come from another related group called plastids, also had about 5,000 genes if we compare it to their corresponding ancestors that are living today. That is, their sister groups. But as mitochondria and chloroplasts have become internalized into a cell, we call endosymbiosis, now they're a little bit like duplicate genes. And in fact, what's happened is a huge fraction of the genes have been lost. Only about 1% retained in mitochondria, only about 2% retained in chloroplasts. A tremendous amount of gene loss, which means that, first of all, these units can never again be autonomous, can never live outside of a plant cell or animal cell. And furthermore, it's caused also some genes to be transformed into the nucleus in both cases. In fact, because the nuclear coding process is much more efficient. So if by chance one of these genes gets dumped into the nucleus or gets duplicated by a gene in the nucleus, then in effect it's much more effective. As a result, some of the proteins that are used to build mitochondria and chloroplasts are also now produced by the nuclear genome. And so we have this process in which you produce this higher order, much more complex cell type. But in the process, you had to both have duplication and degeneration, degradation of the genome of both genomes. Now, the nuclear genome is now dependent upon oxidative metabolism, in which the ancestral nuclear genome looks as though it was not using oxygen for its metabolism. It was anoxic. And similarly, being able to use sunlight to generate power becomes important, but that means that there's certain things that you don't have to do with your genome if you're a plant. So each have become dependent on each other, a codependent, so to speak, in the sense that we're dependent on vitamin C, but now it's, in a sense, two organisms that have become one, or in this case, three organisms, and we think that there's actually more features like this in the process. Now, you've already heard from my student talking about the Bengalese finch. I won't go into this story again, except to summarize what we currently know and we're pursuing. The domesticated species was placed in a context in which selection for singing, in effect, played little or no role in terms of its reproduction. There's some idea that it may have played some role if the male sings an interesting song that the female may produce a little bit more, but in effect, they were selected by their breeders, not for their song, but for their coloration. And this is, of course, done in the laboratory of Kazuo Okanoya in Tokyo. But what has happened is that, as she probably told you, there's been an increase in the song learning, song variation, song complexity, and even in terms of when they tend to sing in the transition from this wild species to a domestic species. Now, as far as we know, there is no major change in connectivity or types of neurons in the forebrain that are involved in either the wild species brain or the domestic species brain. But one of the things that's happened is that it's no longer easy for the domestic species to just produce a normal song without hearing another bird sing. However, the wild species can develop a relatively normal song just by listening to itself. And the way I look at it is that this kind of external suppression of selection, of, in this case, sexual selection on song production, has in effect caused a degradation of, you might say, the template that was innate, that you could listen to yourself produce, and then produce it normally. If it degrades, you have to hear somebody else sing. But it also means that experience, your learning capacity, your tendency to be flexible in your motor behavior now become more relevant features of singing. So many more aspects of the forebrain become more and more important over time for determining the structure of song. It's not that it's taking over production of the song so much as that it's simply the nucleus RA is relinquishing such strong control, such strong constraint on song. But this means that synergistic effects as a result of learning or experience or flexibility all become relevant to the structure of song. Now, in the domestic species, there has never been reselection. That is, this synergy has not been selected because it's more important, although we could imagine that it plays some role. Certainly if these animals were left back out in the wild and there was some advantage to having a flexible song, these might be selected to be much more powerful effects. The logic of this, I have two ways of talking about it. One is that in a bird like the wild finch, the white-rumped munia, if you have a strong innate motor bias for producing a song and a strong innate auditory template, once you produce a song by just sort of spontaneous motor behavior, you hear yourself sing. But now what you've sung, you can listen to and compare it to this bias that you have, genetic bias for what a song ought to sound like. You hear and learn and respond to the difference, and then you modify the next time you sing it, what we call a sub-song. So within the course of a few weeks, a young adult bird will begin with a fairly atypical song and produce a more and more typical song over time. The argument I make is that if you begin to break down these biases, both the sound bias and the motor bias, even with the same logic, the same way of beginning to learn song, you get a very different effect. Now you produce a sub-song that's more variant, so the motor bias is weaker. So the song is more variable as it's produced. You hear yourself sing, and you compare it to an auditory bias. Well, one of them is what you've sung before, but of course you have a weak innate bias. The result is something else has produced this auditory bias. In fact, since you were hatched, all the time you've been hearing other birds sing if you're in a social group, or even if you're around your parents, particularly your father. You're hearing another adult sing. That becomes part of your auditory bias. Now you learn the difference between what you just sung and that auditory bias. You modify your sub-song so it sounds more like that bias that you heard, but now the bias is strongly driven by somebody else having sung. So in fact, there is a series of simulations that we've done. I won't go into the simulation stuff, that argues that this may explain how it is that a song can be passed from generation to generation, minimally affected by the genetics, simply by degrading genetic information. Again, this is a social synergy that's developed, which we talked about, just a synergy within the genome. Now we're talking about, in a sense, a synergy that's social. So here's sort of the depiction I have initially in the wild. Genetics passes on the song. Learning and epigenetic effects and your social experience plays relatively little role. It turns out, play a very minor role, but a fairly small role. However, if you degrade the genetic information, it allows epigenetic effects and learning effects and social effects to play a much more powerful role. And eventually, as this system is completely degraded, radically degraded, where there's almost no bias here, most of the bias is now going to be social. And what you're going to see is this, you might say, offloading of song control onto social processes, social transmission. The point, of course, I'm trying to make here is that this is very much the case for language. We've offloaded much of the structure of our languages onto social groups. A number of analogs are relevant to language. One is that the arousal coupling, that is, we don't have to be in a particular arousal state to produce language. And in fact, it's when babies are in a more relaxed stage that they begin to babble. That phonological biases, that is, laughter may sound the same from culture to culture, but certainly languages don't. Auditory learning plays a role. There's much more distribution in the brain of control of language, so that laughter, sobbing, shrieks, and so on, mostly controlled by basal forebrain structures, midbrain structures, and so on. In us and in most other animals, but in language, huge numbers of areas of cerebral cortex. Now we understand that, in fact, most of the cerebral cortex seems to be involved in language and language learning. Again, a kind of effect, a shift effect that looks a lot like the Finch story. And a whole range of other factors I'm not going to go into. But my point is that there are many features of our language capacity, both behaviorally, cognitively, and neurologically, that resemble these kinds of effects that we see in the bird, and may be the result of a kind of relaxation effect. So what am I suggesting here? That we're in effect a kind of self-domesticated species that may in fact have some degenerate features that make our flexibility possible and make us, in a sense, more influenced by what's going on socially. So that genetic de-differentiation may have played a role. It's rather, it made combinatorial effects and social effects more powerful in us. Now, is there any evidence for this? I won't go into a lot of detail, but let me just give you one example. Very recently, a group down at Stanford has identified something very curious about the human genome. And it turns out that there are some really significant degenerative effects in the human genome. And each of these little blue lines, so each of these is a chromosome of the 23 pairs in the human genome. And each of these little blue dashes represents effectively a deletion. That is where some gene has been damaged. Now, notice how many. There's over 500 here in this picture. Interestingly enough, none on the Y chromosome, because the Y chromosome has almost no role to play in building bodies. But what's interesting is that these are 500 conserved non-coding regions. Now, what is a conserved non-coding region? A conserved non-coding region means that this sequence, the base pair sequence, has been highly conserved. In fact, what we know, it changes almost not at all in all 500 of these in primates. And in fact, in all the primates looked at, and in most mammals, these are highly conserved. So what are they doing if they're non-coding? Well, they turn out to be promoter and inhibitor sections, that is, leading segments that activate or inactivate genes that are downstream of them. That is, they are sites that regulatory proteins bind to activate or inactivate or speed up or slow down the encoding of coding segments of the genome. These are, in effect, regulatory sites. What this tells us is that over 500 highly conserved regulatory sites in the human genome have degraded. That is, expression of these genes has lost their regulatory specificity. This is a remarkable feature. These have been conserved because they must play very important roles in maintaining the structure and the development of the body. Clearly, for them to have all degraded in us says that there is a remarkable relaxation of selection on the specificity of expression of these.
Is this graded in comparison to chimps?
In comparison to chimps, none of these are damaged in chimps. None of these are damaged in rhesus monkeys. None of these are damaged in squirrel monkeys. In humans, these are all deleted or turned into pseudogenes. Because they don't produce proteins, the proteins, there's no change in protein, the numbers and differences of proteins produced, but those proteins are no longer regulated in when and how much they're produced and in which cells they're produced. Massive loss in regulation. Now, my point here is I think that this is relevant to the fact that we are not a eusocial species, but what some people have called an ultrasocial species. That is a species that absolutely depends on the social group, that depends on language and culture to survive and reproduce. But if we've redistributed control onto a large social group, redistributed knowledge of the ecosystem of how to make a living onto our symbolic system, language, then in effect, we become like the Bengalese finch, much more flexible, very dependent upon social learning, and in fact, for incredible aspects, probably, of our development, and particularly of our cognition, dependent upon a social group in a way that we just simply can't survive under these circumstances. But the point I'm making here is this is not a result of complexifying the genome. It's actually a result of degrading the genome. But the point I want to make here is that degradation is a crucial step in making us hierarchically synergistic. So one way to think about this is that we are becoming like endosymbionts in culture. Massive loss in gene regulation, like a mitochondrion, for example, because now we can offload a lot of this onto culture. And as it's offloaded onto culture, it allows a sort of random walk of all of these other effects. So this degeneracy of the human genome is not necessarily making us simpler or stupider, but probably making us much more dependent and codependent upon a higher order, a hierarchic level of control above us. Now, what I want to say, just to end here, is that this means that social transmission, learning and imitation themselves can now become selectively favored, doing a better job of learning social skills, of imitating others, having biases to be conform to others, and so on. All can now be selection to distribute control outside of an individual, to make us better at collective activities. So in effect, we become addicted to being in this large social group in the same sense that a mitochondrion is, so to speak, addicted to being in a larger cell type. But it's important to recognize that the same logic will take place, that there will be a reciprocity. There will be things about the human capability and the human tendencies that aid the preservation of the higher order unit, and aspects of the higher order unit that produce what the lower order units, the now not so autonomous individuals that we call us. So here's sort of a summary. I'll stop with this. I call this a generic evolutionary principle, this duplication, redundancy, degradation, recombination, complementation, synergy, and higher order purifying selection that follows once you get a new synergy. This should be exhibited at all levels. And in fact, this becomes a generic way of talking about how it is that higher order levels are generated and produced. It should also be relevant to understanding all kinds of higher order transitions. And if we think about the Rawlsian story that I began with, it's an argument about how it might have come into being in each of these cases. But it also might be relevant for understanding how to control and regulate or evolve a kind of higher order control in human social organizations in such a way that they're more effective. So as a model for sociology, it might even be relevant. The bottom line here that I think is important is that we need to stop thinking about hierarchic evolution in simple Darwinian terms. We need to be able to think about it both in terms of selection and the loss of selection or the reduction of selection. And that maybe it's the reduction of selection that's responsible for some of the most interesting features in our evolution. So I've gone over time. I'm going to stop there and let people respond.
Wonderful. Thank you very, very much. I'll wait for you to turn off your screen share, and then I will start a question.
Okay. Let me just get to that point. Screen sharing. Now I turn it off. Okay. Let's come on now. Taking a little bit of time here to get it to work. There we
Yeah,
go.
so it goes.
Okay, screen sharing.
To click it. There you are.
Okay, is it back?
Yes, you are back.
Okay.
Yeah, so I guess to start, I was hoping maybe you could summarize a little bit. So you just, in the last slide, you had a list of 6 steps. In the paper that you assigned, there were 4 steps. And I was wondering if you could just name those 4 steps and talk a little bit about how each one leads to the next.
Right. So it's, in fact, we could add more or take away some. So to some extent, it's the general logic that's important, not the particular steps. The general logic is that duplication in some form or another, that is redundancy of some functional consequence, actually reduces selection. The pressure of selection to maintain what we call purifying selection, to eliminate variations, is relaxed. And it's sometimes called relaxed selection. I like that term for that reason. But relaxed selection that increases variation is sort of a standard story in evolutionary theory. But when it's with respect to duplication, something interesting results. Is the relaxation begins to produce variations in functions that are closely related to each other. So the duplicate functions are not totally randomly unrelated to each other. They're related to each other. So if you get what you might call elegant degradation, where a little bit of a degradation takes place, and you have recombination possible by sexual reproduction. And this is one way to say sexual reproduction may have been partially selectively favored because of this consequence. Another feature I haven't talked about here. But because of that, now you've increased the probability for complementing each other's function. So the duplicates, the redundant features no longer being exactly redundant, being partially redundant, can actually play partial supportive roles for each other and cause each one of them to vary more synergistically with respect to each other. By virtue of recombination, if they do so, and a new synergy develops that is actually advantageous compared to the simpler, non-complemented function, then in effect, natural selection will take hold at this higher level and will now tend to increase the combinatorial reliability of that relation. In so doing, this allows loss of some of the original specificity of function in each that's now redundant with the other. And so there will be both simplification at the level of the individual components and, in a sense, greater and greater role for the synergy effect. So this is one way of talking about how it is that, first of all, higher order synergies can show up, can be coordinated with loss of autonomy at the lower levels. And then, in effect, once the synergy is present, there's now going to be selection to maintain that synergy, especially if that synergy is dependent upon things that are outside the genome, social, for example, or between organisms like in symbiosis. So this is an argument about the shift to higher order units of evolution. How it is you can, without some sort of magic mutation that says it just happens, how by regular processes, incremental processes, systems can become, in a sense, shifting their evolutionary, you might say, stability onto what amounts to social or synergistic interaction effects. These are some of the most interesting and important things in evolution. And up to this point in time, we have not had a story to tell how you get there. In the case of Leo Buss's story, or in the case of Maynard Smith and Zothmary's story, we can talk easily about how you protect against them falling apart. As Rawls' story is about how you protect it, how you stabilize your synergy. But it doesn't tell you how you get there in the first place. For us, at a social level, that question, how do you get there from here, is the critical question. how do you move from autonomous selfish behavior incrementally to truly synergistic codependent behavior? I think our species has partly gone that way. That's why we're the way we are. But we have a long ways to go. So that's going beyond what I said a little bit. Excuse me. But the idea is that this is a mechanism that we haven't noticed. And we haven't noticed because we've been thinking about everything in the sort of negative neo-Darwinian terms, in terms of just variation and selection. And all this says is it's more complicated than that. And that in this particular case, for these particular events in evolution, for the very possibility of moving hierarchically, the more complex hierarchical systems, you have to sort of abandon this sort of one way of thinking about it. So it's not exactly a summary that you wanted, but I'm trying to sort of link it all together.
That was splendid. That was splendid.
So I'm open to the question other than yours. You also can drive it.
I suspect. I'm hoping that many of you have read the piece I sent out. I'll be happy to share these slides if they haven't been shared already. And I'll be happy to answer questions both now and online later if they come up.
Sorry, there's a bit of a lag here.
It's all right.
I want to jump in.
It's a lot to take in. So I think the summary, I guess, of how these things can evolve is that the original genes can degrade and duplicate and lower control, and that allows room for the other individuals to get variations of that type of control, and then that leaves room for them to develop things that could be synergistic with each other, and that gets selected for. And is that like the general idea I'm getting? Oh, you're muted. Hold on, I didn't hear you. Your mic's muted.
Terry, your mic has gone out.
Am I back?
Yes, you're back.
Okay, so you've got the story right. The key problem that this is an attempt to answer is how it is that organisms or functional units give up autonomy. Autonomy is something that ought to be conserved in evolution. In fact, replicator selection stories are all about, those that maintain their autonomy and control their own replication are the ones that win. And if you lose it, if you give up your autonomy, give up control, then you're at risk in evolution. This is actually an argument that says, okay, if that's true, and to be hierarchically more complex, autonomy has to be given up at lower levels. How does it happen? Yes, you could imagine some massive magical mutation that simply says, okay, you're now dependent, and guess what? Everybody around you is also dependent. It suddenly happened by coincidence. Now you can develop means to protect the backsliding, so that nobody becomes autonomous again. This is an attempt to say, no, actually, there's a way that you might say, elegantly lose autonomy and elegantly move into synergy. Incrementally, generation upon generation, without having any kind of, you might say mega mutation to make it possible that everybody seems to share all of a sudden at once. So in that respect, it's really an effort to say, stop looking inside the box of natural selection as the whole story. It's bigger than that. And in fact, even think about its opposite, which is what we're talking about. That its opposite is actually a complement. it's not what's not happening in evolution. It's just simply the complement to natural selection. The critical piece of this is that this is not just a logic for biology, not just a logic for genetics. Although what it does say is that the effects, even at the social level, can ramify all the way back down to the genetics. And I think that's what the human story tells us. But in fact, it can also happen at higher levels. And although I haven't said anything about it, I think this is a critical story for talking about the synergies that we call societies, and how they tend to fall into it, maybe in our deep history, but also maybe an insight as to how we're going to move forward.
So oh gosh. All right. So the problem is I have to mute you in order for me to talk, and then I have to anyway. You got at this a little bit with the Gregory Bateson quote. But one thing I was hoping that perhaps you could talk a little bit more about is that you seem to have implied that this relaxation logic is in large part responsible for the modular theme and variation patterns that you see all throughout biology. So could you speak a little bit more about that? Okay, I think you're unmuted now.
No, I'm not.
Yeah, you're good. I can hear you.
You can hear me? Okay. It says I'm muted, but I guess I'm not. So clearly the theme and variation story was really a critical part early on in this story. And the theme and variation, of course, I talked about at the level of regulatory genes and duplication of body parts and variation of body parts, and how developing this hierarchic control of genes, where there's a gene that controls other genes, one gene that controls a suite of genes, and one of those genes might control a suite of other genes. You get large-scale body effects. Why? Because these are about relationships between genes. That is the synergy that's already there that was developed in the development of organisms initially. Now you're, in a sense, got a higher order control of that synergy. And by generating variations of control of that synergy, you, in a sense, you can develop a new synergy of synergies, so to speak. So it's really a critical stepping stone to understanding how this hierarchy of nested relationships, in which the gene duplication effects is nested in regulatory gene duplication effects, is nested in body part duplication effects, is nested in social, or you might say niche construction effects, is nested in social effects, and so on. It basically says that each time you develop this sort of hierarchic control, you produce a new level of individual control that itself can now be part of a higher order synergy. And so you basically got synergies within synergies within synergies. And this is how the whole system is linked. This is why changes in human social organization can actually have these genomic effects that I talked about at the end. It reaches all the way down because this is a whole hierarchy of nested synergy relationships, controlling lower level synergy relationships.
So we have time for one or two more questions. Mel wants to speak.
Okay.
Yeah.
Mel, you go ahead.
Okay. Oh man. so Terry, I read Incomplete Nature, or began reading it last year when I was in a philosophy of biology course, and I got really fed up with the limitations of the neo-Darwinian framework and how that percolated through other sciences. And was reading a lot of Gould and Lewontin and alternative accounts to that. And then I wrote a couple of papers on teleodynamics before it occurred to me to Google search the word and see if anyone else had made that up. And I'm still not certain that what I meant by it was the same as what you had meant by it. And I'm wondering if we could talk about teleodynamics briefly.
All right, am I on?
He's laughing at me.
So can you hear me? Okay, good. So yes, I'm not the only person to come up to coin that term, and it's for good reason. Because it's just simply a way of saying end-directed dynamics. So for anybody that would want to say something about end-directed dynamics, it shouldn't surprise us that that word has been coined. Another word that I thought I coined that I didn't coin was morphodynamics. Morphodynamics was coined in the 1930s, it turns out, and forgotten. It was Weiss who came up with this term initially, and he's talking about epigenesis of organisms, of multicellular organisms. That is that they're producing new morphologies. During development, an organism body generates new morphologies. So morphodynamics was good. It turns out that independently from that, it was used in geology to talk about things like the formation of fluvial plains as a result of the way that water distributes and creates these structures as it comes down out of the mountains onto a plain, creates all kinds of interesting three-dimensional branching structures and so on. So geologists have independently coined the term morphodynamics. So you can find the term in those two different contexts previous to me, that obviously in fields that I didn't pay attention to, the morphodynamics and development. Everybody forgot about it in the 1950s. That word just sort of fell out of use. It was still in use right up to the time I coined it in geology, but it was not a field that I ever read anything about until I said, I better Google the term and see if anybody else said anything before I make any strong claims. I had already used it in my work and then found it in this other stuff. Well, teleodynamics was also used by Dorian Sagan, interestingly enough, in a piece that he wrote. I think it was written after mine, but he didn't pay attention to the fact that I had coined it. I haven't found it in anything earlier than that. But he used it for a totally different purpose. Dorian Sagan was very much interested in sort of the Gaia kind of hypothesis that evolution was directed by purpose and so on. And its use was entirely different. My use was to contrast it with a term that became really ubiquitous in biology, mostly after Ernst Mayr. And that was the term teleonomy. Biologists had long recognized that you need to have purposive like talk in biology. You need to say, so Ernst Mayr talks about, did the sea tortoise, the sea turtle, swim to a particular island and just lay their eggs and swim out? Or did it swim to the island in order to lay its eggs in a place so that they would survive more appropriately, and then swam away so that it wouldn't get in the way? The in order to seemed to be appropriate. But in the 1940s and 50s, it was really important, as it was for Darwin, to get rid of this sort of teleology perspective. These organisms weren't doing it because they wanted to do it, because they had some goal in mind that they were trying to achieve, even though they were trying to achieve something in some other way. So teleonomy was coined by this guy Pittendridge to talk about the appearance of goal-directed activity that could be used in cybernetic analyses. For example, in talking about what a thermostat does or what a guided missile does. It's error correcting, it's deviation minimizing, Maybe we could talk, and so Meyer's thinking, maybe we can talk about organisms as having a kind of program that evolution produced, that's sort of like, you know, a cybernetic program, and that's just acting it out. That there's no real teleology here, that we shouldn't talk about it as teleology. So teleonomy was coined again in the 1950s in order to compensate for that. And people right up till today are critical of the use of teleodynamics because they say, no, don't you want to mean teleonomy? And my argument is, no, we don't want to mean teleonomy. And the argument I given, you might remember from Incomplete Nature, is I say there is another teleonomic system that we could talk about. If you want to say that evolution is just sort of, you might say, frozen accidents of teleonomy being reproduced, you might be able to think about it also in terms of unnatural accidental thermostat. And my example of an accidental thermostat is the Old Faithful geyser in Yellowstone Park. Old Faithful geyser is deviation minimizing because it keeps pressure and temperature within a certain range and then blows off the water in a regular cycle in order to sort of maintain this constancy of pressure and temperature. But that activity plays no role in the existence and the persistence of Old Faithful geyser. Eventually, the heat will decline, the amount of water that goes in there will change, and Old Faithful will change its behavior. The regularity is completely external, as is the regularity, for example, in a thermostat. It's produced by somebody arranging the structure. But in organisms, the regular activity of organisms generates the structure that produces the regular activity of the organism. There's a kind of loop of causality that actually has to do with its very existence. That is not true for Old Faithful geyser, not true for guidance systems and missiles, and so on and so forth. So teleonomy is not enough. It's not enough for biology. We needed a new term. So calling it teleodynamics, a dynamics whose end is the maintenance of that very dynamics. Or autogenesis, which basically says, you know, it's a process, an activity that generates the capacity to produce that process. That's a difference. And so my whole point was to come up with a term that maintained the telos, that is the end-directedness, but said that the end-directedness was about itself in some sense. And that's what's unique about life, and I think it's what's unique about mentality as well. So that's sort of the logic that led to this. And I should say that we came up with this word teleodynamics in discussion. You've been to some of my discussion groups. It took about 6 months to decide that we really needed a term like this, that we really needed to say, no, this is different than what everybody else is saying. This is not teleology in some God sense. It's not making the claim that natural selection, that evolution is contributing to teleology. It's not. Evolution is just honing it and restructuring it.
It's emergent.
Yeah.
Yeah.
So it's a separately emergent kind of dynamics, a teleodynamics.
Can we consider the process of emergent teleology and complexification in terms of like an informational integration?
Clearly it is. Clearly information is involved. In order to have a dynamics that's oriented with respect to its future possibility of elimination, organized with respect to preventing its future possibility of elimination, which is what organisms are about, that state of being eliminated doesn't exist at that point. So you've got to have a representation in some crude sense, information about what is not yet the case, what could be the case, built into the dynamics. So it shouldn't be a surprise that the concept of information or representation is always implicated when we talk about life. Whether we talk about it as genetic information or epigenetic information or mental information. So I think they're critically the same. Telos is about an end that doesn't exist yet, may not exist, could exist, is likely to not exist. So telos is always about, the notion of intentionality. I also play with that term in the book, and it took us also 6 months to come up with the word intention, to say this like intentionality, but it's internalized and it's simpler, it's not mental. The etymology of that, tension, of course, is leaning against or leaning towards. Intention was originally leaning towards something, leaning towards an end, something that's not it. It's not intrinsic, it's always extrinsic, it's always relational. So that's also captures the notion of representations. Representations are something with respect to something that they're not. However you want to think about that, there's always something else. It's always relational. And it's a relational feature between something that's present and something that's not present. And that's, in effect, what drives the whole book. Now, what this discussion adds to that, this discussion is not in my book, for example. This is a totally separate story, right? But it's about the buildup of higher order telos. How lower order teleodynamic processes become complexified and nested to produce higher and higher order teleodynamic relations. Eventually, one of those that we want to explain is what we're doing right now. Right? We want to explain that, but we got a ways to go. There's a lot to it. But you need to have a story about this hierarchic complexification before you can even start.
So one of the, the signature thought moves in everything you've been saying thus far is this absential thing, right? This thing about constraint. And constraint played a really critical role in a lot of the things that you were saying earlier in the conversation. It's the lack of natural selection that's doing these things, or the redundancy, removing constraint and things like that. So I was wondering if maybe we could conclude with some discussion of the role that absence and constraints plays in the story that you just told.
I think you've done a pretty good job of capturing the core piece there, which is the natural natural selection is about the imposition of constraint on a teleodynamic process, a teleodynamic system. Constraints on what's necessary to maintain itself and eventually maybe to replicate itself, and to replicate the information, or to replicate that system of constraints that make the teleodynamics possible. That's what's being replicated. Obviously, the matter and energy gets exchanged, but those constraints maintain. Information is embodied always in constraints, though it's always embodied. Constraints are always embodied in material and energy. They can be passed from one material system to another. And that's, of course, how replication takes place. So natural selection is about the constraints that are imposed that are imposed that maintain the transmission of constraints. But what I've just been talking about is the relaxation of constraints. Not of all constraints, but selective relaxation of constraints. Redundancy produces selective relaxation of constraints. Now, this is true also, for example, in information theory. Redundancy shows that a communication channel is more constrained. Loss of redundancy says that there's more noise in it. So it's a consistent notion with even basic information theory. But here's what's interesting about biology. Biology is about not only maintaining constraint, but also potentially replicating constraints, producing new constraints, producing new individuations of constrained systems, copies of themselves, so to speak. But that in itself is redundancy. A very core feature of teleodynamic processes is that they tend to spontaneously produce redundancy. So the very process of maintaining constraints, of being constrained in how those constraints are maintained, also creates this higher order relationship in which you can have redundancy of constraint. And redundancy of constraint can, under the circumstances like I've described, reduce external constraint, and therefore enable the possibility of synergies to be, in a sense, explored. So it's built into the very nature of teleodynamic processes, the very nature of living processes, that they will tend to produce both constraint, that they're about the stability and the maintenance of constraint, but in that very capacity, there is the possibility, almost the inevitability, of the relaxation of constraint. So in one sense, what you can say is that evolutionary processes themselves, teleodynamic processes themselves, will inevitably have a hierarchic logic to them. It's not because evolution is trying to do anything, or that the evolutionary process itself is teleological. It's that the units themselves have a teleodynamics. And that teleodynamics can produce these higher order effects, including higher order forms of teleodynamics.
Great. Well, thank you so much. That was quite a lot, and I really appreciate it. Yeah, we will be making videos about your talk, and I will be sure to send you links when those videos are completed. And thank you once again for joining us. Does anyone have any final things you want to say before Terry leaves, and then I'll just check in with the class?
Let me just add one thing. Okay. I'm happy to answer your questions online in the future. If you're going to send me an email, very simply, it's deacon@berkeley.edu. Remember my last name, you got it. In order for me to know that it's worth responding to you and not one of the bazillions of others who just write me random questions, mention that you were part of this seminar. Somewhere, either in the email or in the header of your email, so that I know that this is just not some random person from elsewhere, that you've been listening to all of this and it's percolated through your thinking and you've got some criticism or some informational question you want to pose. And I'll be happy to respond. Wonderful.
All right. Well, have a great day.
All right. Well, thank you all. And you've been very patient. I hope I haven't overflooded your stack memory.
I mean, probably, but it's good to get overflooded every once in a while.
Good, good. All right. Well, have a great day, all of you.
Thank you.
All right.